Lecture 11 is about pedigrees and introduces the concept of linkage in pedigrees. A pedigree is essentially a family tree that also denotes phenotype and genotype information. A circle represents a female and a square represents a male. Men are square. A filled circle and a filled square represents affected individuals showing the trait in question. A horizontal line between a circle and square represents a mating. The progeny are drawn directly below the parents connecting to the mating. And so this mating gave rise to 3 daughters and a son. This pedigree shows the inheritance of a Mendelian trait. To remind you, a Mendelian trait is completely determined by a single gene. In this case, the affected state is caused by a dominant allele. You should be able to infer the genotypes from the phenotypes in this pedigree. If I were giving you an exam on introduction, that would be the type of question I would ask. Of course, this seminar is on modern human genetics and there are no exams. However, I can give you an assignment. Assignment 11A concerns linkage of 2 genes which represents a violation of Mendel's second law. Mendel's second law applies to the case of 2 different genes or loci contributing alleles to the progeny. It states that all possible combinations of the 2 sets of alleles can come together in the gametes because the alleles segregate independently. So in the case of the C gene with a dominant big C allele and a recessive little c allele, and the B gene with a dominant big B allele and a recessive little b allele, then you can get gametes with big C and big B, or big C and little b, or little c and big B, little b and big C, or little c and little b in equal numbers. For assignment 11A, I want you to take a piece of paper and draw the Punnett squares for 2 different cases of the dihybrid cross. This dihybrid cross. In the first case, what if the B gene and the C gene are on different chromosomes? What are the genotypic ratios for the dihybrid cross, big C, big B, little b, crossed with big C, big B, little b? So what's the result of this dihybrid cross if the C gene and the B gene are on different chromosomes? The second case is what if the B and C genes are tightly linked on the same chromosome so that the recombination frequency is 0 between them? For both parents, the phase of the alleles is big B and big C are linked together on one chromosome, and little b and little c alleles are linked together on the other chromosome. So what's the outcome, the genotypic ratio, from this cross? To remind you, in the first class, we went over this dihybrid cross from Mendel's P's, and you went ahead and calculated the Punnett square and the genotypes and then gave the phenotype ratio. For assignment 11A, I'm only interested in the genotypic ratio. Please turn in assignment 11A to me at the beginning of class on Tuesday. Assignment 11B is to draw a 3-generation pedigree. Here I just showed 2 generations. So at the top of the pedigree, one grandparent is affected and the other is not. And then for each mating, there are 4 kids. Here I'm just showing 2. And half of the progeny are affected if one parent is affected. No progeny are affected if both parents are not affected. And all the people marrying into the family are not affected. I want you to draw the pedigree in a PowerPoint slide. you can use these circles and lines and squares that I've collected at the bottom to draw the pedigree. So for example, if you just wanted to add in another generation, you could simply move around these squares and circles like that. You can download this slide, Pedigree PowerPoint, from Gaucho Space. So if you go to Gaucho Space, you can see for this week, May 4th to May 10th, there's the slide Pedigree PowerPoint, and you can download it. And then open it. And then make all the necessary changes to make it a 3-generation pedigree. And so on. And when you're done, I want you to save the PowerPoint slide. So this slide, you can save it. Save it with your name. So you can just call it Pedigree PowerPoint your name. And then upload it to your Google Drive. So here's my Google Drive, and here's my pedigree slide, and I can just upload it into my Google Drive. And then we'll use the slide again in class on Tuesday. And so once it's uploaded, make sure you share it with me. Oops. So right, so you can share this with me. Right. Okay. So now back to the lecture. As I mentioned last Tuesday, one goal of modern human genetics is to identify genes that contribute or influence a complex trait. Ideally, we want to look at this pedigree and find some mutation in some gene that co-segregates exactly with the affected phenotype. The problem is that there are 20,000 possible genes, and sequencing all 20,000 for every person in the pedigree to search for mutations is too arduous. So instead, we will examine genetic markers, usually SNPs, that are tightly linked to each of the 20,000 genes. In the simplest case, you can imagine 20,000 markers, one for each gene. So here I have gene X, and there's a closely linked SNP marker Y. We can genotype these markers in every individual in our pedigree. A typical SNP marker is biallelic and so has 2 different alleles. In this slide, I named the 2 alleles 1 and 2. Because the SNP, 1 among 20,000, is tightly linked to a mutant gene X, and let's suppose that this gene X contributes to the complex trait, then we would expect that this particular allele, number 1, would exactly co-segregate with the disease phenotype in our pedigree, whereas allele number 2, which in our example is linked to the wild-type gene X on a chromosome, this would co-segregate with those who are not affected. Thus, we are searching for which among the 20,000 markers are co-segregating with the disease phenotype. That will help us identify genes that contribute to the trait. For assignment 11C is to answer question 12 on the term paper. What diseases are associated with your trait, and pick one. For most of you, your complex trait is a disease phenotype, and so you can just state your complex trait. For one of you, Ramiro, you need to state 1 or 2 diseases associated with, say, abnormal cholesterol levels. In summary, today we covered the topics of pedigrees and linkage, where linkage is a violation of Mendel's second law. On Tuesday, we will perform a simple example of linkage analysis on a pedigree to identify a gene that contributes to a complex trait.